to Chapter 9, lecture video for ecology. I know it's been a while. I'm a little behind because I started research late, waiting to see if we would even be open. So I've been doing lots of stuff in the lab, but I'm catching up now. I didn't really have an outfit to wear for population growth, so I just put on my comfy favorite sea slug sweater that I got off of Etsy that has different sea slug species on it. Because I'm ordering new sea slugs for my tank at home this week, which I'll put them in a video if they survive the transport to my house. So let's get into it. Let's talk about population growth. Okay, let's talk about the ways in which populations grow and how ecologists study that growth and model it and predict it. So first we got to talk about the factors that affect population size and therefore population growth, whether or not the population size is going to increase or decrease. We're going to talk mostly today about birth and death, and then at the very end we'll talk about how you might model immigration and emigration into a population, but mostly today we're talking about birth and death. Birth is going to increase population size while death is going to decrease it. Immigration is movement into a population. It's going to increase population size. Emigration is movement out of a population. It will decrease population size. But like I said, we're mostly going to be talking about birth and death today. Here I have two different types of models for looking at population growth. This one here is geometric population growth. The model of geometric population growth describes population growth over discrete time intervals. And because the pattern of population growth is an upward sloping curve, the rate of population change is going to depend on the time interval being viewed. So you don't, in this model, have a constant rate of population change. So delta n, the change in population size, is going to be greater here than it is going to be at this time interval. And so this model is for something where you're looking at specific discrete time intervals and how growth might change there. With a population that's growing geometrically, the rate of population growth is continuously increasing as the population size increases. And this model can only predict changes over a discrete time interval for which you actually have empirical data. So that's the limitations of this model. Now, the other model that we're going to talk about is over here, and this is the exponential population growth model, which is super important for right now in this time period. This is an instantaneous rate of population growth, and so you have a rate of population growth over the entire time period, and so you don't have to just look at single snapshots. It's not changing, and it's expressed as a proportional increase per unit of time. So R here represents instantaneous per capita growth rate. I will repeat that. R, which is really important, represents the instantaneous per capita growth rate. When R is greater than 0, that means that birth is occurring at a higher frequency than death within a population. The population size is going to increase exponentially. When R is less than 0, which means that there's more deaths happening than births, then there's an exponential decline in the population. And when R equals 0, which means that there's an equal number of births happening as there are deaths happening, then there's a flat line. You have no growth or decrease in the population. One of the limitations of this model that you don't have with this is that it's assuming a continuously accelerating or decelerating rate of population growth as a function of population size. But I would say for what the current situation that we're all living in, understanding an exponential growth curve model is really important. Both of these exponential and geometric growth models are best suited for modeling populations that live in favorable environments at low population densities. So, for instance, during a colonization event of an organism or a virus that is establishing in a new environment, or perhaps an invasive species that has recently immigrated to a new uninhabited area. That's what these are mostly used for. So, let's talk about how it relates to COVID, because it's really important to understand what an exponential growth curve is for understanding how we got where we are and why I'm doing lecture videos. I found two really good articles from the UK on exponential growth. This is exponential growth in the UK. This is empirical data collected by the European CDC, and very clear, very strong exponential growth increase. Although this is only confirmed, it's not the total number of cases, so it's probably even steeper during these first few days, probably because when you see this trend in a lot of places where COVID has spread, because we didn't understand that masks were effective right away, and so there weren't protective measures in place yet, and which is why you see this really steep exponential growth curve. This graphic that they also had in this article is super, super useful, I think, for understanding the effect that even a small change in R can have when something grows exponentially. So basically what you're seeing, so in the article they say that the number of people that are infected from no action can range from 1 to 4. So one infected person then spreads it to 4 people. And so what they did for this graphic is they just took basically like the average of that, which is 2.5. So you can think of 2.5 infected people as the R value for under an exponential growth curve model of no action. So you don't social distance, you just go around, no social distancing, no masks, whatever. You can see how that very quickly over 60 days spreads to 406 people infected. That's a lot. But if you decrease that R value just a little bit to 1.25, it can have dramatic effects on the growth curve of that spread. So if you put social distancing measures in place, make masks highly suggested but maybe not mandatory, then you're only getting 1.25 as your R value, and then you only have 15 people infected. Now let's say you reduce it even farther, you make your R value 0.625, you have social distancing, social isolation, masks in effect, then you only have 2.5 people affected. So I thought this was a really great way of showing how changes in R values, like not a big change at all, can dramatically affect a population's growth rate if it's following an exponential growth model. There's another article that I found that was really interesting that was trying to explain why people were failing to grasp how quickly this could go bad, and it has to do, they use this term called exponential growth bias, that people just kind of are biased to not grasp how fast this can be. So in a lot of people's minds, they thought about the growth of this happening along a linear path, in which case you don't have that many people infected. But as we know, for most viruses, depending on how transmissible they are and their path of transmission, a lot of pathogens and viruses in particular have exponential growth curves, and exponential models are much better fit for predicting their spread. And so you can see with the exponential, way worse than linear. So take a look at those. I put a link to all those on Schoology, so you should look at those. I thought they were really interesting. So those models are really great if you're assuming birth and death rates at the same frequency for all the individuals in the population, but which might be true for viruses that replicate really fast, and an individual viral particle doesn't live that long. But for if you want to take these models outside to animals, for instance, you need to account for age-specific mortality, because not all individuals have the same probability of dying or giving birth at the same ages. And that's where a life table comes in. So you can use them to estimate the per capita growth rate of a population when birth and death rates vary with age. And actually, life insurance companies use these a lot. This is actuarial sciences is a whole field of statistics that's used for calculating things like human life tables. And ecologists have kind of co-opted them to study other animal populations, and plant populations, and other organisms and fungi, lots of things. So this is a life table for people. The first thing that you need to do when you construct a life table is determine what you're going to consider a cohort. So basically, I have to place along the continuum of ages that exist within a population. You have to place those individuals into bins based on age. So for people, you could put everyone born on the same day into a bin, but that's probably way too small and not really that useful. And so for this one, it groups people by age by year. So everyone 0 to 1 age in 2009 was put in there. Everyone 1 to 2 was put in here. 2 to 3 put in here. And then what you can do is you can calculate survivorship for each of these cohorts. So that's the probability at birth of surviving to any given age. And then you can also calculate age-specific mortality. So that's the number of individuals that die during any given time interval divided by the number alive at the beginning of that interval. So I will repeat that. The age-specific mortality is the number of individuals that died during any given time interval divided by the number alive at the beginning of that interval. So you can see various birth and death probabilities calculated here. So, and these are based on actual numbers within each of these cohorts. So humans are interesting because it doesn't look like the probability of dying varies that much. At least within the U.S., we don't have super huge mortality rates, especially if you're looking across a whole population. And this is just a tracking of 100,000, so that's why you see 100,000 even here. This is just a tracking of 100,000 individuals. Anyway, we'll talk about smaller, less complicated examples in the next few slides. So now let's talk about some different types of life tables for how you're going to define a cohort, and then how you define age structure, because depending on the type of organism that you have, it may or may not be that easy to track how long they live, like a single individual, and track its birth and its death. So a cohort or a dynamic life table follows the fate of a group of individuals from birth until death. So this is like well, actually, this is a really good example. So let's say this would be really good for studying an animal that maybe lives in a burrow, where you could tag the pups and then follow the pups around their entire life, like do mark recapture. So you would find them in their burrows. I think they do this with marmots up at Rocky Mountain Biological Lab, and they do that obviously with these ground squirrels up in Tioga Pass. You mark the pups while they're still in their burrows, so you know within a few days how old they are. So you're tracking the mothers as well. And because they spend some time in the nest, you can mark them all. You know their relatedness as well, which is really cool. And then you track them throughout their life. They disappear. Maybe they got eaten by a wolf or something. And then you can track them, see when they give birth, that kind of stuff. But as you might imagine, that's really hard to study things that may not nest or live in burrows or not social. So there you can do a dynamic composite life table. So what this does is it constructs cohorts from individuals born over several time periods rather than just a single time period. So you're kind of building these probabilities from successive generations rather than a single group. And then you can have a time-specific life table, which is probably the most feasible for most organisms, because you sample the population at a certain point in time and obtain a distribution of age classes during a single time period. So if you can reliably estimate the age of the organisms that you're studying by, like looking at their morphology or something, then you can do a time-specific life table and be like, oh, we found 7 deer that are 3 years old, 4 deer that are 2 years old. You can do stuff like that at a single point in time. With the time-specific life table, you're going to assume that each age class, that you sample them proportionately. So there are some assumptions that limit the power of this. And then you're also going to assume with a time-specific life table that the age-specific mortality rates and birth rates have remained constant over time. So there's a few more assumptions that go into this than with the others, but in terms of feasibility, it might be easier if you can reliably estimate the age of an individual organism without knowing the day that they were born. Most life tables in general have been constructed for long-lived vertebrate species, that live up to 10 years. Can you believe these tiny little squirrels live for 10 years? And have overlapping generations, so like people, little baby squirrels. I don't remember seeing any of these, but they live at Tioga Pass. This is a picture I took at Tioga Pass when I lived out in California. It's a high elevation site within Yosemite National Park. You should all go there if you ever get the chance. But anyway, I guess they've been doing these demographic studies of these ground squirrels up there for a while because we're going to come back to them. Anyway, these are different types of life tables that you can have, and some of the limitations of them, some of the life history traits that need to be true for those organisms for this type of life table to work, that kind of stuff. Lots of animal species, though, that aren't vertebrates only live through a single breeding season, which actually makes it a little easier to do a life table. So, for instance, insects are a great example. A lot of insects do not have overlapping generations, so you only have one age class, but you can break them down based on their developmental stages. So in this life table that you've got for gypsy moth, which is an invasive species in the United States, oh my gosh, I have to tell, okay, you probably won't remind me because I go on too many tangents when we meet in person anyway, but the reason that gypsy moth is in the United States is cray, and we don't have time for it here, but I should tell you about it sometime. Remind me to tell you about why we even have gypsy moths here in the first place. Anyway, gypsy moth are an invasive species in the northeastern United States, and so if you were building like a life table of an insect, you would break it into developmental stages rather than like year 1, year 2, because they only have a single generation within a year. So for these, you get values of survivorship from one stage to the next by just observing a natural population several times over its season and then estimating the size of the population at each time interval that you are studying at. So for a lot of insects, you can obtain these NX values by just estimating the number of surviving from egg to adult. So actually this table is really cool. This was actually just collected from a single egg mass of a gypsy moth. So they make these like weird frothy egg masses if you've ever seen them before. There were 450 eggs within a single egg mass. And then this is the number that survived to instars 1 through 3, instars 4 through 7. I had a brain fart about Roman numerals. Pre-pupae, pupae adults, So actually, very few of these eggs actually live all the way to adulthood, to reproductive capabilities. So NX is the number of survivors at each stage. So this is basically time point 0. That's why LX is the probability of survival from that stage to the next. It's 1 because this is time point 0. This is 0.7 because 315 of 450 survived, so that's 70%. And then this would be 57 divided by 315, and so on. And then DX is the number of deaths in each stage. So this is basically this number, that number minus that number equals 135. And so that's the number of deaths. And then the age-specific mortality rate is QX, and that's just that number over that number. Easy peasy, lemon squeezy. We will do this in person in class, because you'll have to do it on the exam, but believe me, I'm limited in my math capabilities, so it will be very simple math that you'll have to do on the exam. What's more important to me is that you know how to interpret the numbers. What would be really cool is if somebody did a study of spotted lanternfly like this, to develop a predictive model of how fast they might spread through Pennsylvania and through Allegheny and Westmoreland County. Maybe somebody should do that for a senior research project. I don't know. Okay. Now what you can do with this life table data is that you can then use it to generate things like survivorship curves. And hopefully all of these look really familiar to you. These are all survivorship curves for like a single species. But they can also be useful for comparing one time point to another time point, different geographic areas, or one sex with another sex. So let's do a little review from Gen Bio 2. Type 1 is strongly convex. The survival rate is high throughout most of the life, with heavy mortality at the end. Humans are a good example of this. A lot of mammals follow a type 1 curve, and then there are some plants, not many, that also have a type 1 curve. Type 2 is a straight line. You've got survival rates that do not vary with age. A lot of birds, rodents, reptiles, and perennial plants will follow a type 2 curve. And then you have type 3, which is concave, and you have really high mortality rates really early in life. So oysters are a pretty classic example of a type 3. A lot of fish, invertebrates, and a lot of plant species have really high mortality early in life, but once they hit a certain life stage, then they have a much higher success rate. And these are some like classic examples of each of the three types, but these are idealized models, and not all species will fit a type 1, type 2, or type 3, which we'll talk about in a second. I told you we were going to get back to these cute little Tioga Pass ground squirrels. This is a comparison of a survivorship curve for males versus females of this species. And you can see that males and females both have high likelihood of survivorship early in adult life, and that generally decreases over their lifetime, but that females have a slightly less steep curve. Females also tend to live to an older age, which kind of makes sense. If they're the ones that are reproducing and contributing to the next generation, it might be beneficial for them to live a little longer and to not have such steep survivorship curves. So this could tell you, especially if you have a population where the females have higher survivorship than the males, it might tell you that they might have a population increase because they're the ones that are contributing to the next generation. But that's not like a direct measure of fecundity. So let's talk about that in a little bit. Now, like I said, the type 1, type 2, and type 3, those are idealized survivorship curves. Not all species are going to fit that. One example that does, this is a Dall sheep. It has a typical type 1 survivorship curve that humans also tend to match, where there's high survivorship throughout most of life, and then as they get into senescence in their older ages, you start to see a big drop in survivorship. The striped plateau lizard, which is right here, this is a typical type 2 survivorship curve. It's pretty constant rate of survivorship across their lives. And then C and B, the painted turtle and the Allegheny Mountain dusky salamander. So this thing lives around where we are. I hope we find it whenever the world is safe enough for me to do ecology field trips again. These two are kind of like a hybrid, combining features of the three different types of at different times during their life cycle. So for the turtle, you kind of see a type 2 and then a type 1, and then for the C, you see kind of like a type 1 transitioning into like a more stable rate of decline into a type 2, but they're kind of like a hybrid of all three. Is the reality of most species is that they're kind of a combination. Now let's talk about net reproductive rate. This is an important thing to be able to calculate if you're studying population fluctuations of a species. So net reproductive rate is the average number of females that are produced during a lifetime of a newborn female. I will repeat that definition. Net reproductive rate is the average number of females that are produced during a lifetime by a newborn female. And in this fecundity table, it's accounting for age-specific birth rates, which is fecundity, B sub x, and then survivorship for each age as L sub x that we saw earlier. And then if the net reproductive rate is equal to 1, females are just replacing themselves every generation. So they only have one of their female offspring reaching maturity and reproducing. If the net reproductive rate is less than 1, females are not replacing themselves. So maybe they are having a lot of males, or maybe none of their female offspring are surviving. And then if the net reproductive rate is greater than 1, then females are more than replacing themselves. So for this gray squirrel population, the R sub O, the net reproductive rate, is 1.4. So in this gray squirrel population, the females are replacing themselves and a little bit more, and so you're likely to see a population increase because of this net reproductive rate over 1. And we'll do some calculations of net reproductive rate in class, in person. And all of this data, these life tables, these fecundity tables, these survivorship curves, they can be used to project population growth. So they can be used particularly maybe in species of risks or ones we particularly care about because they are us, like this human population. You can then use the parameters and all the empirical data that you measure in current time to then calculate things like net reproductive rate or the per capita growth rate, and then extrapolate that out into the future to predict what might happen if things stay the same, if they change, if they decrease. And so that's why this kind of data is really important, is for future projections. So you can see that this graph is using things like mortality and birth rates in human populations by country to project into the year 2100 what the population sizes of each of these countries might be 100, no, oh my gosh, more than 100 years from now. Less than 100, I mean. So one thing we haven't talked about yet that isn't accounted for in these models is stochasticity. And you may or may not have heard this word before. It's used a lot in biology and also in statistics. So stochasticity is a random process, but random in the way that sampling error is random. So I'm asking you right now to recall what you learned about when you learned about genetic drift in Gen Bio 2. How genetic drift is basically sampling error. It is a random assortment of alleles pulled from a population, and that tends to be less diverse than the population it came from. That's genetic drift, is a random process. It's stochastic. And so that's what we're talking about here, is stochasticity that can occur in a population as it grows. All projections of population dynamics, if you're trying to project into the future, are based on probabilities that you calculate from empirical data, but what happens to a single individual in that population is completely independent of other individuals in that population for the most part, unless they're mating with each other. Each individual is independent, and so random processes are going to happen to those individuals independently. And you can account for this stochasticity or these random events through probabilistic models that allow for variation around the mean estimate. Now, I could throw words at you like Markov chain Monte Carlo or Bayes theorem, but this isn't a graduate level ecology class. It's an undergraduate level. So if y'all go to graduate school and become ecologists, you'll have to learn all about Markov chain Monte Carlo and Bayes theorem and all the probabilistic algorithms that ecologists use to simulate this stochasticity. But I'm not going to make you learn about that. I just want you to know that there are probability models that can account for stochasticity in data when you're trying to make future projections or even project into the past. Maybe I'll have you read this really cool paper where they did that with bison. But anyway, you can account for random stuff by allowing some sort of variation around the mean estimate of whatever you use to calculate your parameters from empirical data. Now, you can account for demographic stochasticity, which is variation in birth rates and death rates that occur in populations from year to year, just based to sampling error or stochasticity. So it's demographic is variations in birth rates and death rates. It's obviously not going to be the same every year. And then environmental stochasticity is variations in climate that can directly influence birth and death rates within a population. So maybe some sort of like hurricane or weird temperature fluctuation. You can model that with these probabilistic models that include some sort of variation around the mean. I think these are cool. Anyway. Now let's talk about the real reason I think that understanding these kinds of like population and demographic processes is important, and that's for studying species that are endangered and at risk of extinction. So if you have a net reproductive rate if the net reproductive rate is smaller than 1, that means you have more deaths occurring than births, and a population is going to decline. If that's a species that's in danger, that's a bad thing. And if it's not corrected, that can lead to extinction. So, smaller populations, as you also learned in Gen Bio 2, are more vulnerable to things like demographic and environmental stochasticity. So, genetic drift, like I brought up earlier, genetic drift is basically a random sampling error in the genetics of a population, and the smaller that population is, the bigger the effect. The smaller a population is, the bigger an effect a random error can have on that population. So, the larger the population, and you're taking out a subpopulation of that randomly, that's going to have less of an effect on a population, the bigger it is. But if the subsample you take out of that small population happens to be half of the population, it can have really dramatic effects. And so it can happen through genetic drift or for demographic or environmental stochasticity, and that just makes smaller populations more susceptible to extinction. In previous years of ecology, I've taught a lab where we use this conservation program called Vortex 10, which is, one of these stochastic simulations that you can use to look at extinction processes. It's mostly used for things like large endangered mammals, where you know exactly how many individuals are in the population. You know how many seal pups an individual seal births a year, because you can watch them give birth and tag them individually. So it's mostly, and actually you can see in the vortex that's here, it's mostly large charismatic megafauna that you use this program for. But this is just one example of how you can use these probabilistic models and all of the growth models that we talked about earlier to help save and protect endangered species. Now, I'm going to talk about a species I care about that's endangered, but that we haven't done, that scientists have not done a ton of growth model simulations or anything like that on, but it is important to understand population size. And we have different models for looking historically at a population and currently. So this is a graph of Bombus affinis, the rusty patch bumblebee decline, based on museum records of collected bumblebee specimens, and you can see a really precipitous decline starting in the 90s. They're now critically endangered. It's the only bee in the continental U.S. that is currently listed on the endangered species list. And so this map here displays specimens that were from museums that were collected before 2002. This is the distribution of them after 2002 through 2012, and it's much, much smaller than it was previously, and so there's an average decline of 69% in this bumblebee species. Yeah, not great. But with tools like the population modeling that I showed you, we might be able to help save the species. And I also put on Schoology a link to a documentary that a friends and colleagues, multiple, were involved in producing to help raise awareness for the rusty patch bumblebee. So please watch that. And that's it for this lecture. So stay tuned for the next video, which I'll probably post tomorrow. Okay, bye-bye.